6-Bisphosphogluconate. Undergoes oxidation and decarboxylation by the enzyme 6-Bisphosphogluconate dehydrogenase to form the ketopentose-ribulose-5-phosphate. This reaction generates a second molecule of NADPH, ribulose 5-phosphate. The enzyme phosphopentose isomerase converts ribulose 5-phosphate to its aldose isomer, ribose 5-phosphate, glucose 6-phosphate. G6P is oxidized by the enzyme glucose 6-phosphate dehydrogenase G6PD to form 6-PHOSPHOGLUCONO-beta-lactone. NADP is the electron acceptor and forms NADPH 6-phosphoglucono-beta-lactone. The lactone is hydrolyzed to the free acid 6-PHOSPHOGLUCONATE by a specific enzyme lactonase. 1 molecule of glucose. Formation of 1 molecule of glucose from pyruvate requires 4 ATP, 2 GTP, and 2 NADH. It is expensive. 1,3-bisphosphoglycerate. The enzyme PHOSPHOCLYCERATEKINASE transfers the high-energy phosphoryl group from the carboxyl group of 1,3-phosphoglycerate to ADP, forming ATP and 3,2-phosphoglycerate. In the second glycolytic reaction that generates a compound with high phosphoryl group transfer potential, ENOLASE promotes reversible removal of a molecule of water from 2-phosphoglycerate to yield PHOSPHOENOLPYRUVATE PEP 3-phosphoglycerate. The enzyme PHOSPHOGLYCERATEMUTASE catalyzes a reversible shift of the phosphoryl group between C2 and C3 of glycerate. Mg2 is essential for this reaction, 2-PHOSPHO-acyl phosphate. Any molecule with the general chemical form Rc equals OO2. Anaerobic, hypoxic conditions. Many organisms regenerate NAD by transferring electrons from NADH to pyruvate, forming lactate. Animals cannot convert acetyl-CoA derived from fatty acids into glucose. Plants and microorganisms can. Biotin, a vitamin, an enzyme cofactor involved in carboxylation reactions. Briefly describe the possible metabolic fates of pyruvate produced by glycolysis in humans and explain the circumstances that favor each. Under aerobic conditions, pyruvate is oxidized to acetyl-CoA and passes through the citric acid cycle. Under anaerobic conditions, pyruvate is reduced to lactate to recycle NADH to NAD, allowing the continuation of glycolysis. Dehydroxyacetone phosphate. Dehydroxyacetone phosphate is rapidly and reversibly converted to GLYCERALDEHYDE3-phosphate by the enzyme TRIO, ethanol fermentation. The anaerobic conversion of glucose to ethanol via glycolysis, also called alcohol fermentation. Ethanol fermentation. First, pyruvate is decarboxylated in an irreversible reaction catalyzed by the enzyme pyruvate. Decarboxylase does not involve the net oxidation of pyruvate, requires Mg2 and has a tightly bound coenzyme, thiamine pyrophosphate. Second, acetaldehyde is reduced to ethanol through the action of the enzyme alcohol dehydrogenase, which the reducing power furnished by NADH derived from the dehydrogenation of glyceraldehyde 3-phosphate. Explain why pi is absolutely required for glycolysis to proceed. Inorganic phosphate is an essential substrate in the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Fermentation. Energy-yielding anaerobic breakdown of a nutrient molecule, such as glucose, without net oxidation, yields lactate, ethanol, or some other simple product. Fructose 1,6-bisphosphate. The enzyme aldolase catalyzes a reversible aldyl condensation. Fructose 1,6-bisphosphate is cleaved to yield two different triose phosphates, glyceraldehyde-3-phosphate, and aldose, and DIH. Fructose 1,6-bisphosphate greater than F6P. This reaction is highly exergonic and irreversible. The generation of F6P from fructose 1,6-bisphosphate is catalyzed by the Mg2-dependent enzyme fructose 1,6-bisphosphate, fructose 6-phosphate. PHOS-PHOFRUCTOKINASE1 catalyzes the transfer of a phosphoryl group from ATP to F6P to yield fructose 1,6-B. G6P greater than glucose. Reversal of the hexokinase reaction would require phosphoryl group transfer from G6P greater than ADP, forming ATP, an energetically unfavorable reaction. This reaction is catalyzed by the enzyme glucose 6-PHOSPHATASE. Glucogenic. Amino acids derived from proteins are ultimately catabolized to pyruvate and can therefore undergo net conversion to glucose, alanine, cysteine, glycine, serine, threonine, and tryptophan. Gluconeogenesis. The biosynthesis of a carbohydrate from simpler, non-carbohydrate precursors such as oxaloacetate or pyruvate. Glucose. Irreversible under intracellular conditions, glucose is activated by the enzyme hexokinase to be phosphorylated at C6 to yield glucose 6-phosphate. Glucose 6-phosphate. The enzyme PHOS-PHOHEXOSEISOMERASE catalyzes the reversible isomerization of glucose 6-phosphate and aldose to fructose 6-phosphate, glyceraldehyde 3-phosphate. Oxidation of glyceraldehyde 3-phosphate is catalyzed by the enzyme glyceraldehyde-3-phosphate. DEHYDROGENASE to produce 1,3-bisphosphoglycerate. This is the first of the two energy-conserving reactions of glycolysis that eventually lead to the formation of ATP. Glycolysis would soon come to a halt if the NADH formed in this step of glycolysis were not continuously reoxidized and glycogen converted to monosaccharide units by the enzyme glycogen PHOS-PHORYLASE. Glycolysis. The catabolic pathway by which a molecule of glucose is broken down into two molecules of pyruvate. Hexosammonophosphate pathway, pentose phosphate pathway. A pathway, present in most organisms, that serves to interconvert hexoses and pentoses and is a source of reducing equivalents NADPH and pentoses for biosynthetic processes. Hypoxia. The metabolic condition in which the supply of oxygen is severely limited. In mammals, gluconeogenesis in the liver, kidney, and small intestine provides glucose for use by the brain, muscles, and erythrocytes. In the conversion of glucose to pyruvate via glycolysis, all of the following enzymes participate. Indicate the order in which they function by numbering them. Hexokinase, phosphahexosideomerase, PHOS-PHOFRUCTOKINASE1, triose phosphate isomerases. Enzymes that catalyze the transformation of compounds into their positional isomers. Isozymes. Multiple forms of an enzyme that catalyze the same reaction but differ in amino acid sequence, substrate affinity, Vmax, and are regulatory properties, also called isoenzymes. Mutases. Enzymes that catalyze the transposition of functional groups. NADH, formed in glycolysis, must be recycled to regenerate NAD, which is required as an electron acceptor. In the first step of the payoff phase, under aerobic conditions, electrons pass from NADH to O2 in mitochondrial respiration. Phosphenolpyruvate. The last step in glycolysis is the transfer of the phosphoryl group from phosphenolpyruvate to ADP, catalyzed by the enzyme PYRUVATKINASE, which requires K and either Mg2 or Mn2, produces pyruvate and ATP. Pyruvate carboxylase. Stimulated by acetyl-CoA, increasing the rate of gluconeogenesis when the cell has adequate supplies of other substrates, fatty acids, for energy production. Pyruvate greater than phosphenolpyruvate. Show how NADH is recycled to NAD under aerobic conditions and under anaerobic conditions. Why is it important to recycle NADH produced during glycolysis to NAD? Cells contain a limited supply of NAD and NADH. The oxidation of glyceraldehyde 3-phosphate requires NAD as an electron acceptor. It converts NAD to NADH. Unless this NADH is recycled to NAD, oxidative metabolism in this cell will cease for lack of an electron acceptor. Under aerobic conditions, NADH passes electrons to O2. Under anaerobic conditions, NADH reduces pyruvate to lactate, and is thereby recycled to NAD. Substrate-level phosphorylation. Phosphorylation of ADP. The conversion of glucose into G6P, which must occur in the breakdown of glucose, is thermodynamically unfavorable and organic. How do cells overcome this problem? Cells often drive a thermodynamically unfavorable reaction in a forward direction by coupling it to a highly exergonic reaction through a common intermediate, to make G6P formation. Thermodynamically favorable, cells transfer phosphoryl groups from ATP to glucose. ATP hydrolysis is highly exergonic, making the overall reaction exergonic. Thiamine pyrophosphate. The active coenzyme form of vitamin B1, involved in aldehyde transfer reactions. Two reactions in glycolysis produce ATP. Name the reaction and product, indicate which cofactors participate in where, and name the enzymes. 1,3-bisphosphate is catalyzed by the enzyme phosphoglycerate kinase, in which ADP produces ATP with Mg2 as a cofactor producing 3-phosphoglycerate, and phosphoenolpyruvate is catalyzed by the enzyme Pyruvate kinase, in which ADP produces ATP with Mg2 and K as cofactors producing pyruvate. What are the biological functions of the pentose phosphate pathway? The pentose phosphate pathway produces pentose phosphates for nucleotide synthesis and NADPH, reducing agent for biosynthetic processes. What are the two reactions in glycolysis in which aldose to ketose isomerization is catalyzed by an enzyme? Briefly explain how the reactions are able to proceed without the input of additional energy. G6P aldose greater than F6P ketose, and dihydroxyacetone phosphate ketose greater than glyceraldehyde 3-phosphate aldose. Although both reactions have standard free energy changes that are positive, they can occur within cells B. C. The products are immediately removed by the next step in the pathway. The result is a very low steady-state concentration of the products, making the actual free energy What is gluconeogenesis, and what useful purpose does it serve in people? Gluconeogenesis is the biosynthesis of glucose from simpler, non-carbohydrate precursors such as oxaloacetate or pyruvate, during periods of fasting, when carbohydrate reservoirs have been exhausted. Gluconeogenesis provides glucose for metabolism in tissues, brain, erythrocytes, that derive their energy primarily from glucose metabolism, which catalyzes a reaction in which ATP is produced. Pyruvate kinase, which catalyzes a reaction in which NADH is produced. Glyceraldehyde 3-phosphate, which of the enzyme represents a major regulation point in glycolysis. PHOS-PHOFR, yeast, regenerate NAD by reducing pyruvate to ethanol and CO2.